All right. Thank you, Vijay. Thank you for the introduction. And thank you to Vaishnavi, Vijay, and Vidyanand for inviting me to be a part of this exciting series of talks. I've attended a couple and I've really enjoyed it because I hope, as towards the end of my talk, you will realize that com- the emergence of complex systems requires a fair bit of theory as well, which the embryo somehow seems to know. We don't know how the embryo does it. So experimentally, I will show you a story. I'll talk about a story today, which my group has been working on for several years, and it sort of just came together last year. But this is just the beginning, and we would like a lot more advice and input on what is actually going on that to make an embryo appear the way it appears. All right, so let us start. Um, oh, it won't let me. There we go. So we are all in biological systems. Many of us who work on biological systems use a wide variety of model organisms. So the top row is a series of embryos taken from the C. elegans, Xenopus, frog, Drosophila, and zebrafish. And all of these systems have one thing in common, that they're all single cells. So when an embryo is fertilized, it is a single cell, regardless of what system you work on. Then over time, a series of developmental events occur, some of which we will discuss in today's talk, such that some hours, or in some cases, many months or weeks later, a three-dimensional organism emerges, which looks completely different from each other. So these are all different species, and they look physically different from each other. So developmental biology is an attempt to understand how something that is like this becomes something like this. And there are and this has been studied for a very very long time, and we do have a very good understanding of how things achieve 3D form, but that is still incomplete knowledge. So what is known is that in that one cell embryo, this single cell embryo, the mother deposits, the adult female deposits a lot of information, and that information is in the form of mRNA and protein, which is depicted here in this red. And as the embryo divides to form a multicellular embryo, multicellular means more than one cell, it sort of gives up control to the zygote, to the fertilized embryo, and such that now the zygote's nucleus begins synthesizing its own mRNA and proteins. And those mRNA and proteins dictate the later part of development and all adult life and homeostasis. So this is true across the board for all metazoan species. And the timing at which this shift in control from the early maternal driven development to later zygotic driven development is different for different species, but occurs in every one of them. Now, something else. So this is molecular control. So there is a molecular regime in place, and that regime or who controls that regime changes, and the zygote takes over control from the mother. Now, as this process is happening, something else is also happening, which is this single large cell divides to form multiple, hundreds and thousands of many, many small cells. So this is the beginning of multicellularity, where from a single cell, many, many cells emerge. Again, this is a phenomenon that is common across all model systems, all species, all metazoans, all multicellular organisms. Now, the question is, how does this happen? So we are interested in this problem in the zebrafish embryo, and these are the two movies. One is a very old classic movie, the one that's playing here. Here you're looking at the embryo from the side. What is that this began, I'm just looking backward. The embryo began as one or two cells, and that divided over and over again to make a collection of cells that sits on top of this yolk. Then at some point in time, cells that are sitting up here decide to move, and while they are moving, the shape of the embryo emerges from that movement. And the same process you're looking on this right hand side movie, you're looking at the top view. So you're looking at this part from the top, and you can clearly see that there are many cells emerge, and each cell in the next cycle is smaller than the preceding cell cycle. So this is called reductive cell division. And reductive cell division, again, is a common phenomena. It's not unique to zebrafish. And we wanted to, but it has not been studied in a quantitative manner in any species. So we wanted to study, yes, we know cell size decreases, but how does it decrease? What are the parameters on the basis of which this cell size decrease occurs? So these are again schematics of embryos at different developmental stages. This shaded area essentially is thousands of cells in this area, and the same is true for this shaped area. We will come back to exactly what these stages are a little bit later. But for now, what I want you to take from this slide is that when we looked at the sizes of cells, and it is possible to do cell size estimation in all model systems. We did it in zebrafish using an immunolabeling cell biology approach. So these are confocal images that show you this is what the size of a cell is. This is the size of a cell 1 hour later, and this is the size of cells 2 hours later. So there is a clear visual reduction in cell sizes. We next quantified these cell sizes. I'm not going into details of how we did the quantifications, but we used ImageJ and a size estimation pipeline to estimate sizes of cells of comparable cell cycle stages to come up with certain quantitative understanding of how cell sizes change in zebrafish, change during zebrafish development. So these are the 5 stages of development we picked. The 64 cell embryo, because we can visually count 64 cells and make sure that they are all uniform. From then on, the 1000 cell stage, which occurs around 2.5 hours. Then the sphere stage embryo, the shield, and 3 somites. So each stage is approximately 1.5 hours apart from each other, and they are stage matched. So developmentally, they are stage matched. So if you look at them, and these are all wild type embryos, there is a clear reduction in cell size. And when you see when you plot these cell volumes, and the same is true for cell volumes as well, when you plot the average cell area versus the stage of development, you see a very nice trend which follows a power law fit. And for each stage, there's a power law fit. But essentially, what it means is that the change in area and volume is regulated. It's an ordered process. Now, what we wanted to then see, or the other thing is, when we were doing all these size estimations, because we now have sizes of thousands of cells from multiple different embryos, which are wild type, we could now bin these cells into certain categories. And we chose some arbitrary categorization. One categorization we chose was, we would classify any cell that was 10% bigger or smaller than the median value as a medium-sized cell. Of course, the absolute value of that medium size changes based on the stage of development, but at each stage, you will have a collection of small cells. You will have a category of small size cells. You will have a category of larger cells and a category of smaller cells. Now, interestingly, when you bin cells like this, and these are color coded, the small cells are orange, medium is blue, and large cells are categorized colored purple. What I hope you can see is that, yes, the absolute sizes and volumes change, but the proportions of cells in each of these categories are comparable across developmental stages. So that means that even if an embryo has 64 cells or 1000 cells or 3000 cells based on the developmental time points, in that collective, it has about 40% small cells, about 20% medium-sized cells, and about another 30% or 35% large-sized cells. So there is in that collection of cells, there is a certain size distribution, a certain proportionality that is assigned to each size category. And we don't really know. Part of the larger question is we don't really know how these proportions are set. And we'll again come back to this observation a bit later. Now, why study this process at all? Why is this interesting? Now, to us, this was interesting because cell size is an evolving parameter during early development. And it is possible that since once a cell is 64 cell and it divides to make another tier of 64 cell, in that new collective, there may be no magic cell size numbers. It could simply be that it's a stochastic process and the embryo just decides to work with what it has. And additionally, in several mutation screens, actually in any mutation screen that's been done to date, several cell size mutants have been uncovered, which is how we understand how cell size is regulated. But there are no mutants to date that have been found that actually perturbs cell sizes during the stages that I just discussed. So during early development, we do not know of any genetic process, any gene-driven process that sets the cell size. But how cell and organ size are set and maintained later in development and adult life is a very well-studied phenomenon. Now, if you don't know, so typically to understand how a process works, one introduces mutations. That's the whole. That's a very nice way of to understand how things work. Break it, and geneticists are very good at breaking things by introducing mutations in genes, either blindly or in a directed manner, forward or reverse genetics. But if you don't know what is this cohort of genes controlling a particular parameter, how do you change it? So for cell size, it has been, this is again something that has been known for several decades, that the size of a cell corresponds to its ploidy. So it changes with its ploidy. And this was most famously discovered in plants, where if you have plants, species with, this is a diploid plant, the same species, which is tetraploid or octoploid, the overall size of the plant changes. So does its cell area. But it's not a linear, so it doesn't double or quadruple. It's not a linear increase. And this is again another example to show, another data set to show that the actual individual cell sizes change in a plant that is a tetraploid or an octoploid in comparison to its diploid counterpart. Now, in animal embryos also, ploidy has been used, altered ploidy embryos have been generated. But that has essentially resulted in the conclusion that when you change the ploidy of an animal embryo, it is developmentally incompatible. So those embryos do not survive. And that phenomena has not been revisited. So in animals, we can generate altered ploidy conditions, and those embryos are not viable. But to our advantage, this early process of development that I just discussed, and especially in zebrafish, I will show you pictures in the next slide. Especially in zebrafish, once you perturb the ploidy, those embryos do undergo rounds of division and patterning, and you do get a little larva at the end of the process. It survives for about 3 to 5 days of development. And 3 to 5 days is plenty of time to understand if ploidy causes cell size changes in a zebrafish embryo, what is the actual consequence on development? So we had standardized this process a couple of years ago in the lab, and we can very efficiently generate haploid zebrafish by UV irradiating the sperm DNA and using this sperm, the irradiated sperm, to fertilize eggs. And you can generate what are known as gynogenetic haploids, which lack the DNA component from the paternal DNA, the father's DNA completely, and hence develop as haploids. We had also standardized a well-known technique in zebrafish, which is, and these are things that we custom built in the lab. So you can just take a 50 milliliter falcon, cut off its tapered base, and use that as a container with a mesh. Because fish are aquatic species, you need to immerse the embryos in water. So we made these small holders that you can put in a water bath that is kept at 2 different temperatures and switch the clutch of embryos between these 2 water baths to induce what is known as a small heat shock. And this heat shock is quite mild. It's only for 2 minutes. But what that does is it perturbs the process of cell division transiently. So, and that timing of heat shock is important. So this standardization paper from our group tells you what is the time at which you should heat shock to achieve tetraploidy, but not have this technique associated lethalities in the embryo. So we are very confident that we are achieving a tetraploidy without this toxicity, heat associated toxicity, because we screen out those embryos. So it's possible to generate in zebrafish haploids and tetraploids. So now since we have a condition, we have a diploid condition, which is normal. We have a haploid condition in the same species, and we have a tetraploid condition in the same species. So you can go from diploid either towards larger cell sizes or smaller cell sizes, hopefully. So that is something we wanted to check. And true enough, these are the top rows. This is just again a schematic to show the stages at which we have collected these data sets. So the top row of fluorescence images is the diploid, what is happening normally. This is the haploid where we have reduced the DNA content, and this is tetraploid where we have increased doubled the DNA content. And you can see from these images, these higher magnification images, that indeed in comparison to the diploid, haploid cell sizes are smaller and tetraploid cell sizes are larger at the same stage of development. Now, and this is a quantification of that for both cell areas and cell volumes, where yes, all through development, so at every stage of development, when you compare a haploid cell versus a diploid cell, its average cell size is smaller. So on average, haploids are made of much smaller cells. Tetraploids are made of much larger cells, and that matches with area as well as volume. So I'm going to now call it cell size because at each stage we have done both area and volume calculations. So we now have an experimental condition against which we can compare our normal condition and test the idea that is cell size regulation, or is a particular cell size important for developing normally? And do embryos even have a quote unquote normal cell size distribution? And interestingly, we have managed to achieve this cell size. Remember, I said that there were no mutants which we could use to ask this question. And using our haploid and tetraploid experimental perturbation, we had managed to achieve cell size changes during a time period, which is exactly the time we want it to change. And it precedes a very big developmental transition that results in embryos achieving their three-dimensional form. So let's look at what that is. Okay, now before we get into it, I wanted to share this, I wanted to show this movie. And those of you who have lived in Bombay might be very familiar with it, though in COVID times, of course, this doesn't happen anymore. But this is local Mumbai train traffic. And I've gone to college during these times, like this. And I don't think I could do this anymore. But what I want you to understand from this is movement is extremely complex. As a phenomenon, movement is extremely complex. Because each individual is moving. Each individual is moving as a collective. People trip, they fall, they create obstacles. If you have friends with you, you want to go with your friends. You don't want to be disconnected from them. And you have a goal. The goal is to enter this compartment at the right time before the train departs, right? And if you're unable to do that, what you need to have backup plans put in place. So movement is inherently complex. Now in development, in animal embryos, movement just always gives rise to form. So anything that moves, the purpose of that movement is to achieve 3D form. So again, the same principles of complex movement hold true, which is there has to be coordination amongst self-awareness and coordination with neighbors. You need to be aware not just of who is right next to you, but the overall environment. And you need to have backup plans that in case things went wrong or you are faced with obstacles, what do you do? So if you mapped the movie that I showed you previously, which is completely unrelated to early development, the question still stands that how do people or how do cells get to the right place at the right time to contribute to emergence of 3D form? And that's what we will try to discuss. And each embryo does it differently. So the first one is a zebrafish embryo, this is a Drosophila embryo, and the last one is a frog embryo, three different model systems. And they all do things slightly differently. So there is no similarity, visual similarity between any of these three systems, because a frog is inherently different from a fly and from a fish. So they use the same principles of cell movement to do something completely different and achieve a 3D form. Now, and at the end of gastrulation, you get different diversity of embryos. Now, the the principles, when I talk about common principles that dictate those movements that I discussed, the principles we already know are molecular principles. So there have been a lot of intense work, a lot of beautiful genetic screens that have been done in multiple model systems, and that has essentially given us a molecular framework which is evolutionarily conserved and which all species use to attain 3D form. And these are classic developmental signaling mechanisms. So a common molecular toolkit. So I know you cannot read what is written on these little pictures, but that is the whole point, that we have a lot of information. And this information is conserved. This molecular information is conserved regardless of whether you look at a fish or a frog or a fly or even hydra. And it's evolutionarily conserved, it's hardwired into the genes. Yet you have different species that emerge using that same molecular toolkit. So there is something in addition to cell signaling. So cell signaling executes these decisions. At least that's the idea we are thinking along the lines of. Cell signaling executes these decisions, but there is something in addition to cell signaling which contributes to achieving shape. And once you know what those underlying principles are, maybe even those non-molecular principles could also be conserved. And these could be physical or mechanical principles of development, which is what my group is quite keen on trying to understand. Now, if these principles exist, then all of these species do one thing in common, which is this early series of reductive cell division, where it converts this one cell embryo to generate a mass of cells, and then this mass rearranges to give this normally patterned embryo. Now, we have already generated in our condition, we have already generated embryos which are haploid and tetraploid, in which we know that this mass of cells is incorrect. So the cell size and volume, cell size, area, volume changes in a haploid and tetraploid embryo exactly during these stages when cell division and early patterning is supposed to happen just before gastrulation. So the question is, if you begin migration, or if you begin creating a 3D form with an incorrect mass of cells, incorrectly sized mass of cells, would it matter? And the short answer, and yes, we used the same. This slide is just a reminder slide that we've used these tetraploid and haploid generation paradigms to to address this experiment. And the short answer is, let's first look at the 30 HPF embryo, which is a 1-day-old embryo. So this is what a normal zebrafish embryo or larva looks like. It has, by this stage, it has all the major organ systems in place. It's a free-swimming larva with a functional nervous system, cardiac system, renal system, etc. In haploid embryos, which is this middle panel, or in tetraploid embryos, which is this bottom panel, clearly morphologically, the shape and structure of these embryos is very different from a normal condition. So something did go wrong. Okay, now the question is, what went wrong? So there are two ways of looking at this. One way is that because we changed ploidy, we en masse altered all the genes in this embryo. And we have done several experiments to show that during the early phases that we are discussing, which is depicted on this side, that is not true. So the transcriptome of these embryos are comparable to wild type. So there is not a mass change in expression levels of any gene. And the few genes that are perturbed don't have any direct function in early developmental processes, but that is a list we are very keenly looking at. But my point is that this change in morphology, this change in development cannot be simply attributed to change in expression patterns of genes en masse. And more interestingly, none of these conserved developmental mechanisms pathway, the expression of none of those genes are affected in haploids and tetraploids. So we wanted to look beyond genes to see what else could have gone wrong. So these first series of panels going down the slide are normal embryos, diploid embryos. These are the haploids and these are the tetraploids. So in comparison to these embryos one day later, when you look at events, when you look at the embryos themselves, the first day when you've done the perturbation, they don't look too bad. The only small difference that you will see is that these developmental milestones, so we can focus on this formation of this shield, which is this thickening on one side, that is delayed by about half an hour. So rather than create the shield at 6 hours, and these were very, very carefully stage matched to ensure that we are comparing the right time embryo for the right stage. So we have done this multiple times, and it's not because we are just imaging them half an hour later or 1 hour early. This developmental milestone of formation of this shield occurs half an hour or half an hour to 40 minutes later in both haploids and tetraploids. And by the end of gastrulation, so if you go back, if you remember this movie, once all this movement happened, suddenly the head of the embryo emerged and the body of the embryo emerged on one side. So that the formation of that head on the top and the tail on the bottom part, this axis formation occurs, but the angle at which this axis extends, so this is called axis elongation. So it doesn't elongate quite enough. So this theta angle here is not, it's quite less in comparison to what is seen in the wild type. And again, we have quantified this, so I'm not showing you the quantifications, but this is like an obtuse angle. This is slightly less than what the angle is in a wild type embryo. Now, and furthermore, we have continued to characterize this using a few different parameters. I will draw your attention to two movements that these embryos do. One is once these collection of cells form, suddenly you will find that the cells are moving downwards on the yolk. So the whole collection just engulfs the yolk down. And on one part, on the right side of the embryo, you will see that the cells are trying to move to the top of this ball. So if you focus, there is a shield which goes to the top. So that movement is called anterior migration. So cells are here and they crawl all the way to the top. This anterior migration is compromised in the haploids. And you can assess this anterior migration because there are genes expressed in different domains of this moving clump of cells. And in haploid, these two clumps of cells are mixed with each other. They never quite separate out from each other because one of these clumps is supposed to move ahead of the other, and that movement doesn't occur very well. In the tetraploid condition, this movement is affected, but it is not as badly affected as it is in the haploid condition. If you look at a little bit later at 3 somites, somewhere where I've paused this movie, and the reason I paused it is because now you have to imagine that you're looking at the back of the embryo, the back part of the embryo head on. So these series of images are essentially this live image of the embryo tilted at a 90-degree angle. So you're just facing it head on. It is like this, you have shifted it this way. And the reason this image was acquired like this is because to form the back side, this part of the embryo, cells from both sides of the embryo converge to the midline, towards the middle. And that is called midline convergence. And once it converges to the midline, it then extends its axis, which allows the tail of the embryo to form. So midline convergence has occurred. Now whatever has come to the center will extend along the axis. So these are again very classical gastrulation movement, very famously described by Ray Keller. And in both haploid and tetraploids, this coming together of cells from both halves of the embryo towards the middle is compromised. So these are the two halves. We have just used one gene. There are many, many genes that do this. And you can use all of them, but we have used one as an indicator gene. So there are two lines of cells, and these two lines of cells were on either sides. They were further towards the periphery and they have come towards the center. And the placement of these two lines is much farther away from the midline. So they are supposed to come together. They have not yet come as close as they should. They are still farther apart. So what this tells us is that regardless of whether you shift the size of the composition of this collective to the smaller end of the cell size spectrum or towards the larger end of the cell size spectrum, the outcome on development is the same. So this is something we wouldn't have predicted. So the system actually doesn't care whether it is composed of too small cells or too big cells. What it cares about is that it is not composed of the right mixture of sizes. And once it is not made of the right mixture of sizes, the eventual developmental outcome seems to be a common problem. So did you, I want this to be clear that there is a preferred or an optimal condition. Now that optimal condition, when you deviate from it, can result in two different outcomes because the direction in which that deviation occurred is different. But rather what we see is that whether we deviate towards a smaller sized cell collective or a larger sized cell collective, to the embryo, the outcome is the same. Okay, what it knows is that this isn't a normal cell sized collective. That is the information the embryo takes from this process.
Sreelaja, there are a couple of questions.
Sure. I think, maybe I will take a break right now. Yes.
Okay. Chandra, can you go ahead? Please unmute yourself. Chandra, you there? Okay, I don't know if he can hear me. I'll just read out his question. So he's asking if the time for division is independent of the cell size, then one would expect the proportion of the sizes to not vary with time. Is that right?
I don't know what he means by type of cell division, but we will discuss cell division towards the end. So the timing of cell division is not different.
He's here. Maybe he can ask. Chandra, go ahead.
Yeah. So what I meant was the division, the interval between subsequent divisions, if that is independent of the sizes, yes, then whatever the size distribution you start with will, that's one way to just have the same proportions maintained. I'm assuming, of course, symmetric division, like equal size, daughter cells and a lot of things.
Yeah, so we have calculated cell division times in haploids and tetraploids, and they are comparable to diploids.
Okay.
Yeah, so cell division is not we will discuss this towards the end again, but that was also our first guess, and we checked it, and that's not changing.
Okay.
Next is Debashish.
Okay, hi. So I have this small question that do you really have got the distributions of the cell sizes along the development? I suppose you have the information, but yeah, do you have the, for example, the distribution functions? The histograms?
Yeah, I showed the distribution plots of these cells of different sizes. So these are the actual cell sizes of the cells that are quantified. So from every embryo, we have quantified around 900. So these are the total numbers of cells at each stage that we have quantified. We have more cells to be quantified as well, but this is the current numbers of cells that are. Is that what you're asking?
Yeah, maybe that's just a different kind of representation of the same data. But
yeah. So when you plot it as a histogram, this is not a normally distributed data. So you will not get a peak at one. So it is a non-normally distributed data, and we have plotted this in different ways. Okay. So we felt that this was a better way to show the spread of the data, that there is still an average behavior of cells, but there are of course some outlying exceptions, like some cells are bigger here and some cells are smaller.
So I was wondering if you have the distributions, then with at different times, can they be collapsed? Do they have the same behavior, or they qualitatively change along the time?
So I'm not understanding the question exactly what you're asking.
Yeah,
so,
no, it's fine. So
no. So we have looked at this data. We have looked and assessed this data in different ways. So what we have done is, using our confocal slices, we have calculated the cell areas. Now that cell area falls across a distribution. That distribution is not normal. That data is not normally distributed.
Yeah.
Now, to understand what is this mixture of cells that we are quantifying, we have decided a category of cells. So the category we picked was 10% smaller or larger than the average value of at that particular stage of development. Those cells we categorized as medium-sized cells. Anything smaller than that is small. Anything larger than that is large. And within these three categories of cells, we find a conserved distribution across developmental time.
Okay.
So the absolute area values change. But if you pick a category, I'm sure if we had picked a 20% plus minus, we would have gotten the same distribution, the same spread in the categories of cells. But this is one categorization of the mixture that we have done.
Okay, thanks.
Okay.
Next one by Vaibhav.
Okay, aren't the cell size basically decided by the morphogenic gradients rather than just simply the number of chromosomes that are there inside the cell? Otherwise, what are the morphogenic gradients basically doing?
No, so morphogen gradients are not just deciding cell sizes, they decide fates of cells as well. So the primary role of a morphogen or any signaling molecule is to decide what that cell becomes. And there are, so clearly in our condition, you still make a head, the embryo still makes a heart. So morphogen gradients decide where the heart, where the liver, where the pancreas, where the eyes, all that, where the muscles, all of that will form. They don't generally tend to decide how big the eye will be or how, what should be the individual cell ka size in that organ. So that is typically decided by growth factor signaling. And again, when we looked at these kinds of signaling processes, they did not come up to be differentially regulated in the conditions we are testing.
Sandhya?
I think Sreelaja partly answered this question, but I still want to clarify. Can the defects that you see in a one-day-old little zebrafish arise from total number of cells and not just the size? Because in a sense, when you change the ploidy, even though you say the gene regulation by transcripts has not changed, nonetheless, clearly ploidy changes have been known to have a lot of effects.
Correct.
Historically. So I'm just wondering, could it be just you don't have enough numbers? That is, small changes in rates of cell division could add up by the time you become 24 hours, and therefore you don't have enough cells to do the movements.
Yeah. So that is precisely why our entire story is not on a 24-hour embryo, because by 24 hours, there will be many domino effects of what ploidy change has done, including reduction in cell numbers, maybe some cell death, etc. So what we have done is we are exclusively focusing on a regime of development which is early. So you divide multiple times to a very large extent. Up until 6 hours or 10 hours, we have quantified the cell division rates, and we show that is not changed. And therefore, we are reasonably confident that at 10 hours, maybe if you ask me, at 24 hours is a haploid. This is a question that has come to me every single time I've given this talk. If you ask me, is a 24-hour haploid embryo made of lesser numbers of cells than a 24-hour normal diploid embryo, the answer is most likely yes. But that's because by 24 hours, many other things have gone wrong. So today's discussion is exclusively 64 cell, 128 cell, 256 cell, 512 cells, where you can count the cells and you can track the cell division times. So we are reasonably certain that the haploid embryo is not dramatically composed of lesser numbers of cells in the time regime that we are studying it.
Vaishnavi?
Hi, Sreelaja. Yeah, very nice. I was just wondering if some of these effects are to be attributed to cell size. Would it be possible to test and to delineate the effect of the direct role, rather, of cell size on these stages, developmental stages, without possibly including the confounding effects of varying ploidy? Because that's how you're achieving this, right? Is it possible at all to do it experimentally?
Yes, it is possible. And we have done it. So, that was your question comes at a natural transition point in the talk. So,
great. Okay.
If Vijay doesn't have any questions.
I think there's one more. Sorry.
Naman Mishra, go ahead.
Hi, Sreelaja. I wanted to ask if this cell size dynamics is different in protozomes and deuterosomes, or is it same in all the organisms?
No,
Same
it's different. So um, I mean, it's different in the cells that, in the sense that a lot of it depend upon nutrient signaling, including in metazones. But there is also an effect of crowding. Largely, it is nutrient driven. But in systems like the zebrafish or the Drosophila, for instance, nutrient signaling does not play that big a role in deciding whether a 3-hour zebrafish embryo, what is the cell size going to be for a 3-hour zebrafish embryo. But it might play a role in lower organisms.
Okay, you can go ahead.
Thanks, Vijay. All right, Vaishnavi. And with that question, we did exactly that. So ploidy was a necessary tool. We couldn't have addressed this and obtained the kind of insights that we do have without using ploidy, because we didn't know which gene to target. Now, but having found this result, the logical question is, all of this could be because we changed ploidy, and can we try to do the same thing in a ploidy independent manner? And that is what this next set of data is for. So we developmental biologists or all experimentalists do things both ways: perturb, show the same thing happens; rescue, and show that the process can be reversed. So we have done both. And we'll first discuss this recapitulation of this defect in a non-ploidy driven manner. So what we have done is we have deployed embryos. So this entire data set is on only deployed embryos, no ploidy change at all. And we have additional experiments, backup experiments, to show that the experimental perturbation we are doing does not change ploidy, that is data I'm not showing currently. So this is what a deployed embryo looks like, which is treated with a vehicle, and deployed embryos treated with a particular drug combination. This is a standard drug combination that is used and very well established. We had used this drug combination at doses which were much, much lower than, and for much shorter duration. So this is a very acute treatment for about 3 hours, from 3 hours to 6 hours. So cells in embryos, so if we can go back to this movie, so we started treating embryos somewhere around this stage, and we stopped the treatment somewhere around this stage when the shield had just begun to form. And then we are asking, once you treated cells for 3 hours in this window, how does the rest of development occur? So during the crucial phase of cell migration, the embryos are not exposed to drugs. So we want to make a perturbation and ask, what is the consequence of having made that perturbation? So clearly you can see that even with this short exposure regime, and we have done it again for 1.5 hours as well, because in the next slide, we have a much shorter regime, half shorter regime. So the cell size does increase. So this is deployed. These are deployed cells whose sizes have increased. This is quantified in these graphs here. And they tend to show similar gastrulation defects. So this is the axis, the midline convergence defect beginning at the time point of axis elongation. You can see that the two sides of the embryo have not quite come together yet. And this is again shown by gene expression panels on this side, where this is what a deployed embryo's midline convergence would look like. The two rows of cells come towards the midline, towards the center. And these two rows of cells are displaced much farther apart. And these are indeed deployed embryos, because we have FACS and chromosome spread data to show that the ploidy of this condition has not changed. Now the other condition. So since we could use a drug combination and increase the size increase the cell size in a zebrafish embryo, we use this exact same regime in a haploid embryo. So remember, haploid embryos are made of smaller cells. So we wanted to ask, if we subjected a haploid embryo to the same drug regime and increased its cell size, would it have a consequence on development? What would its effect be on development? And that is this series of experiments. So these embryos they have a double hit. They are haploids and they have been subjected to drug. So they are not very happy subjects, but it sort of gets the point across, which is haploid embryos they have smaller cell sizes. Haploid embryos treated with this drug combination, the cell size sort of reverts back to normal. It's different, but it reverts back to normal. And this axis elongation, this midline convergence and axis elongation defect that were part of the original haploid condition are also partially rescued. I'm saying partially rescued because these embryos are not completely wild type. And we have seen with drug treatments, you can see this in this picture as well. There's a slight ruffling so that the haploid embryos tend to be more sensitive to drug exposure. So much so that we have treated the haploid embryos only with a 1.5-hour drug exposure rather than our 3-hour drug exposure. Because even at 1.5 hours, we are able to achieve this increase in cell size in both diploids and haploids. And in diploids, it recapitulates our aberrant condition, and in haploids, it tends to rescue our phenotypic outcome. Now, all of this is leading up to me telling you that what is going wrong is whatever is going wrong is hitting the migratory process that achieves this three-dimensional shape of the embryo. So no matter what this collection of cells is, no matter how you have achieved that collection normally or in our experimental condition abnormally, these collections of cells have to now move. And the movement of these cells is what dictates the eventual shape that emerges. And there are many ways in which we have looked at it. All of it is live. And in fish, it is possible to do it. It is very labor intensive, but it is possible to do it. And one type of experiment is depicted here. So when you have a multicellular embryo, in this case, it's a 16-cell embryo, you can inject one of these cells with a tracer label, and all the descendants of these cells will carry that label. So you now have a mosaic labeled embryo. So you have a clonal condition where in a salt and pepper manner, some cells are labeled in a largely unlabeled background. And now that you have a way to see these cells, you can trace their individual behaviors while they are moving. And this is what this data set is. So this is a normal wild type embryo in which we have generated these clones of cells. And we are now, we have live time-lapse imaged them starting at gastrulation when we could orient the shield. So we know that these cells are supposed to go towards this part of the embryo where the dorsal shield is, where the two parts are coming together. You cannot see the part that's behind the screen because we have taken a side view. And you will see that some of these populations of cells are merging towards the dorsal sheet. Some are going further down because the embryo needs cells all over the anterior posterior axis. Now, if you see the same in haploids and tetraploids, cells do migrate. It's not that they are not migrating, but the end result of this migration, the end position of these migration and their trajectory. So if you just compare the lines at the end of the movie, and in both of these haploid and tetraploid condition, the cells are supposed to go towards this side. That's just how the movie was acquired. You will see that they converge dorsally, but there are cells that are straying off and doing strange migratory movements as well. They are just not getting to this dorsal part as efficiently as they could. And when we quantified these parameters, so each colored dot is a different embryo from which cells have been quantified, and we are showing the distribution of these cells for 4 different parameters. So we calculated the total distance a cell has traveled and the speed at which it traveled. We calculated the total displacement. So that is a total change in net position from point A to point D. And the tortuosity. So tortuosity is distance over displacement. If it is 1, it means that they traveled in a straight line. And what we find is that in comparison to diploid embryos, these clonally labeled cells in a haploid and in a tetraploid. So these are cells migrating in a haploid embryo, cells migrating in a tetraploid compared to cells migrating in a diploid. These cells travel the same distance. So there is no difference between the distance they cover, and they seem to be traveling at the same comparable speed, but they are displaced less because the net displacement, the net change in position is less for both conditions. And they do so because they seem to be taking more tortuous paths. They wander more. Now, one way again, so like we did for the drug condition, one way to check whether how much of this is intrinsic behavior, how much of this is the cell's own behavior versus the influence of its environment, we can put these cells in a different condition, in a normal condition. So you could take a diploid embryo and one of your altered ploidy condition, in this case a haploid or a tetraploid or a drug-treated embryo, label them differently, take both groups of cells and put them in a diploid host. We chose a diploid normal condition because we wanted to see if we gave these cells. So a haploid cell in a haploid environment doesn't have the option of migrating in a normal environment. Everything is abnormal in that embryo. So we wanted to give haploid or tetraploid cells a normal environment and see if they manage to migrate properly. And again, this was done live, where we generated a mosaic clone of cells, a mixed embryo containing cells from both these groups, and traced their migratory process. And that is this kind of, so we end up acquiring these kinds of movies where again you can trace the individual migratory routes of cells. So in diploid, so the red is diploid. In both conditions, the red cells are diploid cells. The green cells are either the haploid condition or the tetraploid cells. And you will see that overall, the red cells do tend to migrate closer to the midline. So they are supposed to migrate on this side where my cursor is, whereas these green cells are always slightly away from the midline. Now, when you quantify this cell behavior, what one finds is that you have different parameters. Again, if you we have analyzed this data in a few different ways, and we chose these 4 parameters because we felt that they represented the basic principles of migration quite well. So a haploid cell or a tetraploid cell in a diploid environment tends to migrate less. So overall, it covers less distance. It tends to be slower. So it covers less distance, it is slower, and therefore it is of course displaced less. The net change in position from point A to point B is much shorter. Now, and so that explains why we always found our green bunch of cells why we always find this green group of cells displaced further towards the end of the movie. Now, and they tend to, but one parameter that is indeed rescue is a tendency to wander. So cells don't, there are some outliers in this data, but on average, if you look at the bottom of this graph, cells don't take very complicated routes. So one thing the environment seems to be able to enforce on the collective is don't wander from your trajectory. So these haploid and tetraploid cells don't wander because their environment doesn't allow them to wander, but they are a little bit retarded in their movement. They are slower in their movement, and that is reflected in the less distance they travel and their consequent shorter displacement as well. Are there questions at this point? I'll get into what actually may drive this phenomena. Okay. So I will. So once we figured out that there were differences in how these cells are migrating, and some of these parameters are changeable based on the environment, what we wanted to ask was, what is actually different about movement? Why are these cells moving differently? And our in-depth analysis of cell migration phenomena originated from that question, though we have not managed to successfully answer that question. All we can tell you in the next 2 slides is that the migratory behavior is actually indeed different at a single cell level. So we can again do that because we generated these mosaics where the membrane of the cell is marked, and we have used a transgenic line, which I've written phalloidin, but that's not correct. What we have used is a utrophin GFP transgenic line in which actin is, the endogenous actin is marked. So it's not a drug-treated condition. In these conditions, what we have done is you can idealize the cell as a circle, and from the circle, we have plotted the length of the emerging protrusion and the direction of the emerging protrusion. And we have, to be able to compare across multiple cells and across multiple embryos, the direction of migration is towards dorsal, which we set as the 0 point. And from then on, you're checking the protrusion placements and length around that 360-degree angle. So this is what the movies look like. This is a normal DIC movie with the, I mean, this is a DIC movie where the transplant is shown and it's live and healthy. We can image this for hours. You can, using the red channel, you can just look at the membrane, which allows to tell you where the membrane actually extends. And the green tells you how much of that membrane contains actin in it. The protrusion contains actin in it. And if you look at, if you do similar analysis in the haploid condition, where the cell size is indeed smaller, you will find that the protrusion dynamics is perturbed, because all of a sudden there are fewer protrusion, they tend to be smaller, and there is some actin in those protrusion, but they're not very long. If you look at the tetraploid condition, or in this case, it was a drug-treated condition, these are fairly large cells. These cells do extend protrusion. Those protrusions do contain actin, and on average, the protrusion length and numbers are much more than what we find in a diploid. So again, here, and this is quantified both in terms of just the membrane itself and membrane plus an actin containing protrusion. So the data is mirrored both ways. Whether it's just looking at membrane protrusion or actin containing membrane, the data stays the same. So this arrow down here is the direction of movement, dorsal. So for all cells, this has been normalized to the direction of movement. And the plot is essentially 45-degree angle ka sectors, where we have plotted how many protrusions and of what length has emerged from that part of the cell. And you will see that in a normal condition, the cell tends to extend, diploid cells tend to extend protrusion that goes away from the direction of migration. Okay. Now, we'll come back to why this is problematic, but this is what we see. We'll come back to why this is problematic a bit later. In haploid cells, when the cell sizes are smaller, the protrusions are sort of all over the place. There is no preferential direction in which there is an absence of protrusion, which could aid in movement. And in the tetraploid condition or the drug-treated condition, here, this is a drug-treated embryo, a diploid embryo whose cell size we have increased by adding a drug. The protrusions are way more in number, they are much longer, and they are all over the place. So there are the entire profile of both haploid smaller size cells and larger size cells completely change. So we believe that this indicates that there is a directionality, but it is not a directionality we would expect. So it's not like we are like we know for cells in culture that it extends protrusions in the direction of movement. It of course extends protrusions in the direction of movement, but it sort of seems to feel what it is leaving behind also much more rigorously than where it is going. So this is a phenomena that we are quite keen to understand, because it is a little counterintuitive from what we would expect for migrating cells. So actin protrusion dynamics is also perturbed, and this may be one reason why the cells have trouble getting to where they should be getting to. Now, I'll do a quick recap of what I told you. So what I've showed you is that these early cell divisions in zebrafish embryos, starting from a single cell to generating a multicellular zebrafish blastoderm, which can then undergo cell movement, follows power law scaling, and it decreases with time. It's a very, very nice regimented decrease over time, both in area and volume. We are able to generate zebrafish embryos where the cell collective is differently sized. And from this differently sized cell collective, embryos develop abnormally. So embryos do care what the composition of the size of this cell collective is. And what I have shown you, and I understand that this doesn't fully answer the question, is that when the sizes of cells in that collective changes, what we're essentially changing is the ability of cells to move coherently as a group. So you are changing cell interactions amongst neighbors, and when they are unable to interact optimally amongst themselves, they tend to migrate aberrantly, and that aberrant migration manifests as a developmental error of pattern. And that is, these migratory problems arise also largely because the actin protrusion dynamics of that cell, whether it is a small cell or a large cell, is different from that of a wild type cell, a wild type normal diploid cell. Okay. Now, besides the fact that it is just an interesting phenomena, why is this important to understand? So, fundamental knowledge is one part, but there is also an added component of function, because organs are multicellular and there is a cell size optima for each organ, a homeostatic cell size optima for each organ. In our conditions, what we find is when cells are made of, organs are made of two small cells or two large cells, what we're essentially changing is the function of this organ. So a rudimentary way, we are looking into this right now. That's work that's led off from the first part that I discussed. This heart, the heart in a haploid embryo or a heart in a tetraploid embryo inherently functions differently. So there is cardiac arrhythmia in these fish. We are pursuing this with transgenic lines and markers, which will allow us to study this phenomena in greater detail. And one direct way this maps to is that in cardiomyopathies, the ploidy of the cardiomyocyte changes, either because the individual has hypertension or is diabetic. The modal ploidy of cells do change. It's also known that modal ploidy of megakaryocytes, which are stem cells that give rise to platelets, they change in a diabetic individual. So these are resident stem cells in the bone marrow, megakaryocytes, and they're modal ploidy. They are polyploid cells. So they contain, the modal ploidy is something like 16 copies of DNA, 16 or 8 copies of DNA, and that shifts to 32 or 64 in diabetic individuals. And it is known that these extra ploidy megakaryocytes or excess ploidy megakaryocytes, from them immature platelets bud off very quickly, and those immature platelets are cleared by the circulatory system. So there is no platelets in circulation, making these individuals prone to bleeding disorders. So ploidy and its regulation of cell size does matter to overall functioning of an organ. And we are interested in trying to understand how individual cell sizes shape organs, either during development or during regeneration. And we would like to approach this not just from an experimental perspective, but also from a theoretical or a modeling perspective to understand how can one, what is this information that a collective of cells needs a priori for a 3D shape to emerge, considering that all signaling pathways, whatever executing mechanisms are needed to execute this decision, exists in place. And then some of those models can be tested in zebrafish, because we have established this model which is testable for predictions. The other, I think I'm nearing the end of my presentation. So the other thing is not just the heart, multiple organs in the embryo seems to be compromised. So this is the gut. So the gut tends to undergoes these peristaltic movements, and the wave propagates from the from the back end of the gut to the front end. So from the front, the wave emerges and then it retracts, and then it emerges and it retracts. So you can see that the gut in the haploid and the gut in the tetraploid, the peristaltic movement is also perturbed. So not just, again, this is also a condition where the cells are bigger or smaller, and this is a second organ system in which function is compromised, and something we are looking to study in further detail. Now, in addition to all of these, so I had a question at the very beginning. So when we were seeing all these perturbations, the overarching question in our head is, how does the embryo, forget haploid and tetraploid conditions, how does a normal embryo generate a collection of cells of differently sized, it's a heterogeneous group of cells, but there is still an optima. So how is this optima set? How does an optimal collection of sized cells emerge? And one way could be by regulating cell size. We again don't have an answer to this. We are in the process of trying to find an answer or a process to solve this question. But first pass, what we looked at was, when we are establishing a collective of differently sized cells, are we changing cell cycle dynamics or cell division rates? And the short answer is no. We have looked at 3 or 4 different time points in the embryo, all of which is before the cells have begun to migrate. And just to explain it, so if you follow any one of these dots, they will split into 2 because that's an interphase nucleus that will undergo mitosis, split into 2 dots. So we trace the mitosis to mitosis duration for multiple cells across multiple developmental time points, and they don't seem to be different. So even if cell division rates play a role, it's not a very obvious role that it is playing. The addition thing is, so besides cell division rates, what decides the size of a cell collective? How do we know neighbors influence cell sizes? Because when we picked cells from a haploid or a drug treated or a tetraploid embryo into a diploid condition, some aspects of aberrant cell migration were recovered. So cells didn't wander so much. So clearly neighbors can influence individual cell migration, but to what extent? Am I influenced by neighbors 2 cells down or just near neighbors? Or I'm always influenced by near neighbors, but each influencing the nearest neighbor sort of sets in place a behavior for an entire row of cells. And I said that there are proportions of cells of small, medium, and large category. Do these proportions of cells of a particular size matter? And is there a spatial map? So is there a particular place in the embryo where it refers to place majority of smaller size cells, or a particular place where it decides to place larger size cells? So is there a spatial heterogeneity to this size collective? And is it random? Is it predetermined? Can we, in a model, can we flip these potential spatial organizations of the shape, sizes of cells, and see whether the eventual shape that emerges, is it different? So I'm going to show this short video. You've all probably seen it. Not probably, I'm sure you've all seen it. This is a very fascinating phenomena of murmuring in birds, murmuring or collective behavior in birds. Sorry, I don't want to. Can you hear me?
Sreelaja, we can hear you, but we can't see you.
Yes.
So this is murmuration in birds. And the reason I show this.
Sreelaja, we couldn't see the video.
You might have to share your screen rather than the window, Sreelaja.
Yeah. We can hear the audio, but not see the video.
Oh, how do I show the...
You may have to share your entire screen.
Ah, okay. I don't want to show the... I mean, I don't want to play the audio. I do want to play the video, though. So let me stop the share and I will reshare the other screen in which I'm looking at this movie. Ah, there it is. Can you see the screen now?
Yeah.
Can you see the screen?
Yeah, we can. We can.
Okay. So this phenomenon is very well known. It is extremely well studied as well and very fascinating. So these are individual collectives of cells. These are individual collectives of birds. These are individual birds moving in a collective. Sorry, and they tend to make these very interesting dynamic moving patterns, which are very complex. And what is understood is that I want to switch back to my keynote. What is understood is that the okay, can you hear me again?
Yes, we can, but we are still seeing YouTube, not your slides.
All right.
Yeah, now you're there.
Perfect. So I played that movie because this collective behavior of birds, of a flock of birds, is a very well-studied phenomena. And that behavior emerges from a few very, very simple principles. One is don't bump into your neighbor. The average speed of birds in that flock is regulated, and there is no real leader. There is no single bird or group of bird that leads. So these are sort of self-emergent systems. So what I want to the way we are thinking about these things right now is maybe early embryos, and in biology such self-emergent systems are known. So ant behavior, for instance, is a self-emergent property. And in early embryos, the way we are looking at it is that maybe this emergence of a collection of cells and the conversion of that collection into a 3D shape could be viewed as a self-emergent phenomenon, because you can the complexity of the process and the fact that there is not a single gene or protein or RNA or collection of proteins or RNA that seems to be doing it, makes us lean towards the hypothesis that these could be self-emergent properties. And these self-emergent properties, a haploid embryo or a tetraploid embryo still makes a zebrafish embryo. So this is a conserved set of rules that the system follows. In one case, it results in a normal-looking embryo. In another case, it still results in a recognizable zebrafish, but its shape is completely different. So if you want to develop normally, if you want a 3D form to develop normally, you need to begin with individual properties of a system that is wild type. And if you change these properties, you still make a 3D shape, but you don't make the correct 3D shape. And we would like to probe the idea that maybe early developmental biology, early embryos, the principles of developmental biology that early embryos use could be self-emergent properties, and we are keen to understand what the rules of this system may be. The work that I discussed, I'm going to end my talk here now, because after this I'm going to just be speculating and it would be good to discuss this. The work I just showed is done completely by Triveni. Towards the end, for some gene expression quantitative PCRs, Rahul helped her with it, and Asfa helped with the cell volume quantifications and cell size, cell migration parameter assessments. But the work was largely fully done by a very talented graduate student who's now a postdoc in Princeton. And I would also like to acknowledge the funding agencies that allowed this work to be done. One was TIFR DAE and Wellcome Trust DBT for the fellowship that we used to fund this work. And I will end my talk here and take questions if there are any more.
Yeah, thanks Sreelaja for a very nice talk. There are a couple of questions. Sonali, would you go ahead, please?
Thanks, Sreelaja, for that highly interesting and very engaging talk. I have a very simplistic question.
Sure.
Now, what do you think will be the difference in gene expression, since I think in terms of genes, in a diploid cell versus a haploid or tetraploid cell or embryo? After all, only one allele is expressed in a diploid cell, and haploid and tetraploid cells or embryos are still alive. Maybe I just missed something, or you are going in the direction of emergence. So
we did look. So we did look at gene mRNA levels we did whole transcriptome analysis to look at mRNA levels in haploids and tetraploids at particular stages of development and compared it to diploid. We don't see the overall levels changing, and we were, the way it was explained to us by people who study dosage compensation. So my group doesn't study dosage compensation. It was explained to us is that if two alleles are making two copies of a gene, and if a single allele is making a single copy of the gene, in the relative pool of RNA, its expression would still be comparable. So what it points to, the dosage compensation group says that what it says is that these are mechanism of dosage compensation is robust in these embryos. So we have not perturbed the dosage compensation process. The single allele transcribes 2 copies rather than a single copy, and that is how it could be maintaining levels of genes. But we have not focused on gene regulation to a large extent because we are shifting we would like to probe the physical aspect of this phenomena in a little bit more detail. So it's not like we're not looking at the gene expression pattern, but we're not focusing largely on it. We're looking at physical principles rather.
Okay. Thank you very much.
Sure.
Vaishnavi?
Hi, Sreelaja. Very nice. I might have missed this, but could you comment on what you think is the mechanism underlying aberrant actin protrusion in these differently sized cells where you looked at different ploidies?
Yes. So there is, so again, because the actin protrusion, actin in the membrane and the propensity of a cell to extend protrusion is also dependent on the membrane area. So it is quite likely that because the area of the membrane is less, the surface area of the membrane is less, and I think there is some work on this already. The area being less, the tension on the membrane could be different. A large cell with a large surface area, the tension on that membrane could be different from a small cell with a small surface area and a regular diploid cell with a normal surface tension and surface area. So I think it is membrane tension dependent presence or absence of a protrusion, and then therefore the presence or absence of actin in those protrusions. Does that make sense?
And yeah, I mean, I think it does. So I guess you then try and see if there is indeed a change in membrane tension in cells. Exactly. And you're not attributing it to changes in gene expression, like you just answered, I guess, right?
No. So I think it is happening on a scale because these processes are so dynamic. They are dividing and they are moving. So individual cells may not have the time or the wherewithal to change transcriptionally everything or over and over again, because these are very, very fast processes. So they divide. So the early part, the up until the 3-hour or a 4-hour embryo where it's making thousands of cells are dividing every 10 minutes. So
right.
They're not spending enough time in interface to actually do transcriptional based regulation. At least that is my viewpoint. I may be wrong. But what is obviously changing is the physical aspect. Things are shrinking rapidly, very, very quickly. Places where you didn't have neighbors, all of a sudden you have neighbors from all sides, top and bottom. So that will influence how an individual cell behaves. And therefore we think that even with the membrane protrusion, all the conditions that you saw for the actin, it is surrounded by normal cells. It is a single cell or a group of cells in a wild type environment. Despite that, it is not able. It doesn't. The aberrant actin property or membrane protrusion property still emerges, which means that the tension is to some extent dictated by the size of the individual cell. The direction in which those protrusions and whether that cell ends up where it is supposed to end up is decided by the entire environment. So it is mostly, I would be very surprised if it wasn't mechanical tension or membrane tension driven. And we are checking that right now.
Cool. Thanks, Sreelaja.
Chandra?
Okay, so taking the bird flock analogy forward, so here there has to be some sort of communication channel between the neighboring cells, right? If anything of that sort to happen. Yes. So one could be that there is surface bound receptors and ligands that come in contact with each other, or there is something that is diffused in a very local environment and being sensed and responding similarly.
Right.
So one can one look at like the correlation between the activities of like the movement of the neighboring cells, like if you tag a bunch of cells which are actually next to at least within a sort of small radius,
yeah,
and see the correlation in the movements, would you see sort of less correlation in the movements in case of the tetraploid and haploid as opposed to the diploid one?
So,
I mean,
So you're asking about some near neighbor analysis in a movie where we have done tracking. So does a cell retain contact with the same set of neighbors over time, and does it change in when the size of the cell changes? So the short answer is we don't know, because all of the trackings, all of the analysis that you saw was manually done, because we are not a computational lab. It's a lot of work to manually track and trace not just the migrating cell, but also the neighbors around it. But we are going towards a more automated or machine driven, for lack of a better word, way of analyzing this data, because analyzing single cells or 20 cells or 40 cells or 100 cells is not the same as tracking the behavior of the entire collective. So what we actually want to do, and we had begun these experiments and then very quickly realized that if we label all the cells in the embryo, very naively we thought that if we labeled it all, then we would be able to trace movement. But that's not true. It just is a lot of noise. But we do have movies where we can, with reasonable accuracy, map migration of a particular cell, and maybe for 10-15 minutes the migration of all its neighbors, and then see if it changes. Because during gastrulation, cells do switch neighbors, and that is partly how they decide where to go and at what speed to go at. So cells switching neighbors is not unusual. And that is especially true when cells divide. So one thing I didn't mention is, up until gastrulation, cells divide a lot, because you are generating this mass of cells that need to be rearranged. But once gastrulation begins, once this collective migration process begins, mitosis doesn't play a very big role. The cells are dividing, but if you prevent mitosis during gastrulation still proceeds. You will get a smaller embryo, but gastrulation still proceeds. So you've already made this bunch of cells that you want to rearrange. But if a cell does divide and make two cells, the daughters inevitably move away from each other. So in some of these mitosis movies, when the screen that is in view right now, in haploids and tetraploids and in diploids, once a cell divides, the daughter moves away. The two daughter cells move away from each other. The near neighbor boundary is only respected if you're not dividing. If you divide, it moves to different neighbors. But that also is, sorry to go on about this, because this is something we've been thinking about for a very long time. It is also possible that the act of cell division and the fact that you now have the opportunity to change your neighbors might lead to seeding phenomena. So you may now have seed cells that have come from somewhere else and will tell the collective how to go. They're not leaders necessarily, but they've come from a different environment into a new environment, and they may influence how that host environment behaves in that embryo. But that tracing we have to do.
Yeah. So I was about to suggest that if you just look at like all the pairs of a bunch of pairs of cells at the t is equal to 0, and see what is the distance between them, and look at a later time and see what is the sort of distance between them. But then I completely forgot that there is cell division, and this actually throws everything off. Yeah. And whatever cell you start with, it might not be there because it would have divided at some point.
But because it is zebrafish, there is a way to get around it. Fish are absolutely nice. So the one way to get around it is you use a time maturing GFP, time maturing label. So it's called CAEDA, and you can have a tracer. Once the tracer is a particular color at time 0, the descendants of that cell which carries a tracer, over time the fluorophore color changes. So if you have different colored cells over time, you know where those different colored cells originate. So there is a way to do it. It's not, it can be done. We have not done it.
Okay. Thanks a lot. Nice talk.
Sure, thanks.
Sandhya?
Nice talk, Sreelaja, actually. It's nice to see the full story together. You sort of answered this question, but I'll still ask you. I have a couple of things in this direction which I wanted to ask you. In your mosaic transplantation experiments, did you put your diploid cells in the tetraploid or haploid environment, or was it only from the tetraploid and haploid back into the diploid? And as you said already in response to Vaishnavi's answer, some of that actin dynamic seems to be encoded by the cell itself, who the cell is.
Correct.
And so this would actually sort of tell you how much is the nearest neighbors, as you said in your talk, going to control the direction of movement. Because it seemed from looking at your movies that really the direction of movement was largely unperturbed in any of these cases. And the actin dynamics is a purely local phenomenon.
Yes.
And so that's what I was interested in, what happens when you put the diploid cells into these other manipulations. Or perhaps I missed it. Maybe you said that.
No, you didn't miss it. So we did not do, it is on the cards to be done, because we also are interested in how much is this autonomous, how much do neighbors influence it. So we are going to do it in all permutations and combinations. But we didn't want to put all of that in here. And additionally, we were already very concerned that we are dealing with quote unquote an abnormal condition. So ploidy, people are kind of wary of ploidy, but it has its advantages and disadvantages. So we didn't want to assess normal cells movement in an abnormal condition because the condition is abnormal. But to answer a fundamental question like the one the actin data poses is, how much of the migratory or protrusion behavior of a cell is intrinsic to its own identity? That question, to answer that question, we will be doing these reciprocal transplants. So that is on the cards. Your second question was, what was the second question?
It appeared to me by looking at your movies that the environment actually did not. Yeah.
Correct. So the overall direction doesn't change because cells are still moving dorsally. Now, that is also a largely unanswered question, that why do cells move towards dorsal? Why do cells go to the top? So when the collection of cells are engulfing this yolk, they involute and go upwards, right? And then once they're reaching upward, as they're reaching upward, both of these groups are converging towards the center. So there is a movement of cells that go up and then they are converging together towards the midline. It is as yet not known why. It is known how cells reach dorsal. So there is fibronectin, integrin connections, there is Wnt signaling, there is BMP signaling. But why do a group of cells choose to go towards the middle? Is not. And we have looked for morphogens that are expressed from the midline that will attract the cells from both sides towards it. That is partly why I have worked on this problem a little bit before, which is my PhD work on chemokine signaling in gastrulation in zebrafish. So the idea was that we might be able to figure out that is there a point along the embryo, is there a zone along the embryo's midline where there is something like a chemokine, let's say, being expressed, and that allows cells that guides their movement towards the midline. That is not true. What the chemokine rather does, it ensures that near neighbors migrate together. Perturbation of the chemokine signaling essentially decouples two groups of migrating cell, but those two independent groups still move dorsally and anteriorly. So why are they moving dorsally? We don't know. But it could be driven by the overall geometry of the embryo. So zebrafish embryos are spherical. And once that spherical symmetry is broken by the formation of the shield on one side, which is a dorsal organizer, that might be sufficient to trigger a physical movement towards one part of the embryo. That is a possibility.
Okay, Sundar?
Hi, Sreelaja. Nice question. Nice talk. I have a question. sort of following up on the answer that you just gave to Sandhya. So you said that, cells sort of move towards the midline, right? Most of the cells. So some of the cells move towards the animal pole, some cells move towards the midline. And at least those cells that move towards the midline are known to be planar cell polarized, right? So, and I'm wondering, like, how much of the aberrant cell movements that you see in a tetraploid cell or a haploid cell is due to a lack of polarization versus a change in cell size. Probably both are interlinked somehow, but I'm guessing, have you looked at polarization specifically?
So we have looked at all components of the Wnt planar cell polarity pathway. So that is data that I did not show. So in addition to the transcriptome data, in which no Wnt or planar cell polarity pathway, planar cell polarity components came into picture. Okay. None of them were differentially regulated. We have individually looked at something like 13 or 14 genes of the PCP pathway. None of them are affected. But that is not to say that they may not be affected at the protein level. The data we have is for RNA. But PCP mutants, so the planar cell polarity pathway has been very well studied by several zebrafish groups, non-zebrafish groups as well, but in gastrulation, Professor Leela Solnika-Kretzel's group in Washington has studied it very well. The phenotypes we see are not quite what they see. Okay. And since our preliminary data did not reveal PCP components, we don't think it may be involved in this process.
Okay, cool. Yeah, thanks a lot.
Sure.
Okay, I don't see any further questions, so then we can end the talk here. Thanks, Sreelaja, for a very nice talk.
Thank you for having me, and thank you for all the discussion and questions.